Welcome to the Ash Cloud. I'm Ash Sweeting. Today we are joined by Yaniv Altshuler, who is using decentralized AI algorithms that are ubiquitous across nature to predict the efficacy of methane-reducing feed additives based on the cow's microbiome. Yaniv developed these mathematical equations during his work at MIT and recently founded Meta AI to scale this technology.
Method that we developed basically allows us to find automatically, in a scalable way, thousands and thousands of genetic patterns that are guaranteed to have some phenotypic meaning.
Genetics has its own set of equations.
We discovered that there exists a set of mathematical equations that are ubiquitous in the sense that they are guaranteed to emerge in any data. Back then we thought any data that represents human activity. So financial data, blockchain data, cyber data, mobility data, social networks. We saw and we were able to show that in each of these data sets, these exogenic ubiquitous properties emerge. Now, the reason this is so important is that now we had the mathematics of data and we can use it in order to find patterns of interest and predict behaviors of individuals and so on.
Solutions need to be dynamic.
Because the microbiome constantly changes as it reacts to changes in the food, weather, and so on, certain feed additives that worked really well at one given herd may stop working, or the other way around. Feed additives that didn't work so well can suddenly become very effective. So we take a fresh sample every 3 months, and of course, we don't need to sample the entire herd. 15 cows from the herd contain enough material to be a good representative of the herd.
Data-driven solutions have an impact.
In terms of numbers, we can triple the efficacy of these feed additives. This is actually the bottom line results.
Yaniv, thank you very much for joining me today.
Thanks for having me.
So AI, there's a lot of talk of AI in the media in all areas, from both a good perspective and a less good perspective. Can you talk us through AI in agriculture and where you see the opportunities and where you see the challenges?
Yeah, sure. So I think that currently we find ourselves in the middle of a revolution. And this is, of course, I think something that should already be quite obvious for everyone. But what we see is that things that were considered quite impossible or even were not even part of the discussion are suddenly becoming something that is feasible. And very soon, some of them might even be turned into, a commodity. So, of course, we are all using chatbots and are witnessing the effect this has on our everyday life. But specifically in agriculture, what we're doing now is switching from a very traditional field that basically utilizes best practices and notions that existed pretty much unchanged for thousands of years into things that can be personalized, that can be dynamic, that can be data-driven and AI assisted. And of course, I'll love to give some examples and of course talk about what we do specifically.
Okay, well, if we just take a little step before we jump into that, and for people who, AI is everywhere. People are talking about AI this and ChatGPT and how it can be used in medicine or space or warfare or anything. And if we take a step back and just for the people who don't really understand it, what actually is AI? How does it work in layman's terms, not in the full details of it? If
Well,
that's okay, if that's possible.
yeah, so basically AI is mathematics. It's a set of algorithms that can solve very complicated problems. So, for example, for quite some time, decades, AI was mainly used in gaming. There are very interesting insights or even theorems that have to do with games. For example, chess, right? So we all know that there was a point in time where Deep Blue was able to win against Garry Kasparov, and it was really a great milestone. But at the end of the day, this algorithm, this AI machine, was using a very basic algorithm from the 60s called A star. It's a generic algorithm that can basically play any game whatsoever, as long as you represent it in the form that A star can comprehend. Then it will just play it, and provided enough computation power, it would eventually win. So it was just a matter of providing more computation and some better heuristics to evaluate the different boards, and then just using the same algorithm from the 60s, and allowing it to suddenly win. So what's quite amazing about AI is that it's very non-linear. With the increase of more computation power and more storage, it really displays quite a remarkable exponential or even a double exponential trend, which means that and the thing is that people are really bad at thinking in exponents. So people may say, this thing doesn't progress anywhere. It's still quite dumb. Yeah, no serious improvement. And then, and it's the same exponent, but suddenly, oh my God, I can speak with it. It passes the Turing test. It wins against chess. It can pass the bar exam. But look, we're still in this exponent, and things are even going to get pretty wilder, I suspect, in the coming years.
So you're using AI to try and address livestock methane emissions. So what does that involve? How do you do that? Where do you get the data sets, and how does that, how does talk us through that whole process?
Yeah, so maybe before that, I can maybe give a little bit of background about my work and my research, and then this I think can help understand what we do today. So I'm a researcher at MIT. My PhD was in computer science, focused on basically decentralized AI. I was working on my PhD 20 years ago when decentralization and AI were much less trendy than they are today. And back then, we were asking ourselves, so what actually is decentralized AI? And the answer that we came up with was basically the ability of a system to work in dynamic conditions with a lot of uncertainty, in a way that doesn't involve a central controller or a central algorithm. Now, this was a bit high level, but you can think of a swarm of drones that are launched in order to find someone who got lost in the woods. And you have thousands of drones looking for their rescue target. And you don't really want to have some central commander to tell each drone where to go, because it doesn't scale. And also, you don't want to assume that all the drones would exactly do what you think that they are going to do, because some may malfunction or have some sensor issues and so on. So what you want is to have some local algorithm, something that works in the microscopic confined area. So a single drone, maybe some rules that govern the way drones break symmetry if a small number find themselves in the same area. And what you want is that through those local rules, a macroscopic swarm scale behavior will emerge. So this was the topic of my PhD. I also published a couple of books on these topics. And the reason it's so fascinating was that it led us deeper and deeper to the fundamentals of data and the fundamentals of AI. Eventually, during my postdoc at MIT, working with Professor Alex Pentland, we were asking ourselves, okay, so what is data? What can we say about data that is completely agnostic to what this data represent? And what we discovered was quite remarkable. We discovered that there exists a set of mathematical equations that are ubiquitous in the sense that they are guaranteed to emerge in any data. Back then we thought any data that represents human activity. So financial data, blockchain data, cyber data, mobility data, social networks. We saw and we were able to show that in each of these data sets, these exogenic ubiquitous properties emerge. Now, the reason this is so important is that now we had the mathematics of data, and we can use it in order to find patterns of interest and predict behaviors of individuals and so on. And for about a decade, this is what we were doing. Very interesting projects out of MIT, working with the various government agencies and large banks and so on. And then several years ago, during COVID, we were wondering, perhaps these mathematical equations can also be applied to genetic data, and specifically to the microbiome. And this was somewhat of a leap, but it was very easy to test. And we basically took a bunch of data, microbiome data, and this data was sequenced. So it's basically billions and billions of strings representing nucleotides. And what we witnessed was that these equations also emerge in this kind of data, which, if I wanted to really be, pompous, I would say, I could say that, basically this is the language that evolution is writing genetic material with. And quite remarkably, there are a lot of similarities between the language that is used to write genes and to write genetic material and the English language. I can, of course, talk hours and hours more on this aspect, but this was the foundation of what we're doing now, because we basically discovered that we can find patterns, genetic patterns, without knowing what we're looking for. So this is the basics, and I'll explain why this is so important. You've probably heard of tests that are designed to detect mutations that cause cancer and so on in humans. And of course, this is very important because if someone finds out that they have this mutation, then they can undergo a surgery or take some medication and so on. But this meant that people spend probably, a decade or so looking specifically for this kind of mutation, trying to find the cause for this specific mechanism that goes wrong and so on. The method that we developed basically allows us to find automatically, in a scalable way, thousands and thousands of genetic patterns that are guaranteed to have some phenotypic meaning. Namely, we can basically build a machine that can harvest from a large collection of genetic material thousands of biomarkers. Some of them may be associated with some terminal disease. Some of them may be associated with the efficacy of drugs. Some of them may be associated with some trivial thing that doesn't have any meaning. But the thing is that this is done in an unsupervised, unbiased way and at scale. And so this was quite a lengthy introduction of the science behind what we do, but now I can actually tell you what we do. We use this technique in order to predict the efficacy of feed additives for cows, basically allowing farmers to use the best feed additive for their specific herd at any specific point in time in order to achieve the maximal impact of these additives. Bottom line, farmers are being given the opportunity to tremendously decrease their carbon footprint, decrease their methane emission, and increase their yield, either body mass in beef or dairy yield in dairy cows, by simply optimizing the digestive systems of the cows.
There's so many questions and so many points that you just raised. Many of them would take us deep into all sorts of rabbit holes, which we probably want to avoid. But thank you. That's fascinating, that whole thing. The fact that these equations exist throughout nature, nature continues to fascinate me the more I learn about it and the more I hear about it. And in terms back to the microbiome, so you've got the fact that the rumen microbiome or the soil microbiome, there's a range of organisms there. It's very different. We think of genomes, and we think of ourselves or the actual cows, so mammals, large animals, or even plants, but which are all eukaryotic cells, so they have nuclei. And then when you start to look into the microbiome, there's combinations of eukaryotes plus prokaryotes, which don't have nuclei, so their DNA and the way they divide is somewhat different because of that, and the way they metabolize. They don't have mitochondria, they don't have a lot of those organelles, so they're quite different in terms of their function. So how does this work in that complex environment? Because there are literally billions and billions and billions of processes going on every second within that rumen. So how does talk us through that, please?
Yeah, so you touched on a very important point. Every microbe is a tremendously complicated machine, but not only that, the microbes forms ecologies. And what researchers have been trying to do for quite some time was trying to find the bad microbe, the microbe that makes everything goes out of tune and so on, or find a good microbe and just add it to the mix. But in fact, because we have an ecology, because we have network effects, this is actually not the right way to take. What we would have liked to be able to predict is what happens when a certain composition of microbes are present. And this basically is a very difficult problem if you want to solve it through the modeling of the functionalities of the microbes, because there are hundreds of thousands of microbes. And in the rumen, there are, let's say, 1,000 different microbes. So it's 1,000 from hundreds of thousands, and this explodes combinatorically. There is no way that you can actually model what's happening in the ecology of 1,000 different species out of hundreds of thousands. Leaving aside the fact that about 90 or 99% of all the microbes in existence are completely unknown to science. So you're trying to sequence the microbes only to end up with between 1% and 10% of the material actually being used, and the vast majority of the data just being thrown away. However, this is not what we do. We take a purely data-driven approach. We don't even try to identify who are the microbes that are taking place in the rumen. Instead, we're treating the DNA sequences as our objects and are looking for patterns that coexist in microbiome samples. Sub-strings of nucleotides that appear together in a too coincidental way, which is a non-formal way of saying that are basically very unlikely to just spontaneously appear in genetic material vis-a-vis the equations of our social physics theory. So in other sense, we are using these sets of equations that we know govern the way evolution converged in terms of encoding genetic material, and using them to find anomalies. And anomalies are basically functional units that sort of make sure to appear close together. So using this way, we can actually find, let's call them decentralized genes, functional genetic units that may origin from different microbes. They don't necessarily have to be in the same single microbe, meaning that some phenotypic property occur when this and this and this microbe are together, or microbe number 3 can be switched with microbe number 7. So this is why it's so difficult to just solve this problem with traditional ways, but using this method, this can actually be done.
So in terms of the practical approach and building this, you obviously need to know what the rumen microbe is, and you need microbiome is, and have samples of those and have them analyzed. And there's short and long chain sequencing, so there's different types of sequencing and which ones work. And then you'll need some data about, because I imagine you mentioned feed additives, but that's anything that the animal eats, because if they're eating the pasture or the hay or the silage or the concentrates are also a huge part of what's driving that microbial activity. So it's the whole diet plus any specific additives that are in there. So how does that all work? How does that all fit together?
Yeah, you're absolutely right. So with respect to what the animal actually eats, we assume that the farmer would feed the cows with whatever they prefer to feed it, most likely dominated by the financial considerations, and we cannot influence that. So we are actually taking the basic feed out of the process, because this is something that is given. It's changing, but it's given. And what we care is the influence of the feed on the microbiome. Now, the microbiome is affected by many other factors as well: the weather, pathogens that may be introduced to the herd, and so on. But of course, many things affect the microbiome. The microbiome basically is our sensor. We assume that everything propagates to the microbiome and changes it, but this is the final step that govern how much milk will be produced, what is going to be the fat content and the protein composition of this milk, and so on. How much methane is going to be released, which basically means the efficiency of the digestion process. So the microbiome is the engine that may be influenced by many things, but we tap directly to the microbiome as the last point in the chain. And regarding the additives, when I say feed additive, I'm talking about the abundance of products that were recently introduced to the market, very good technologies, and lots of capital and work that have been invested during the past decade in the development of those additives that are being added in very small quantities to the feed, intended to improve and optimize the digestion of the cows. And there are many different technologies. Many people are talking about seaweed, and there are products that are based on extract from garlic and citric oils and so on. Now, the thing is that these products are really, really great sometimes, but their efficacy is very inconsistent. They may work great, or they may not work at all, and anything in between. And it's very difficult to predict what their efficacy is going to be. And this is exactly what we provide. We allow farmers to predict which additive they should be using. Now, to your first question, we have spent more than 2 years collecting what is today the world's largest data set of cows' rumen microbiome. We've sequenced more than 1,000 microbiome samples from cows, and we compared it with thousands and thousands of measurements of productivity and digestion efficiency and methane emission that we've collected over the course of these 2 years. So basically, we have the technology that can detect biomarkers in the microbiome. We've collected a very, very large data set containing microbiome samples and labels, and the labels are exactly that. Which feed additive worked here and didn't work there, and so on, because this measurement actually encompasses cows that were untreated at all and were used as control group, and cows that were given feed additive 1, 2, 3, 4, and so on. So this allows us to basically collect this efficacy data for the feed additives and then contrast it with the biomarkers, training our system that can now be given a fresh sample of microbiome and output a prediction. This feed additive is going to work really well for this herd, this feed additive not going to work so much, and so on.
There's growing amounts of data, and obviously this space is an area that needs much more research. So there's more, most trials lead to more questions than they do answers. But there's data that the efficacy of feed additives will change in the same animal over time. Obviously, different additives have different modes of action and impact the microbiome in different ways. So that's also a variable. How does your data kind of look at that, equate for that, and manage that side of things? And do you need to get extra samples over time to understand that? Is that something that you can do from, it's more predictable than that?
Yeah, no, you're absolutely right. Because the microbiome constantly changes as it reacts to changes in the food, weather, and so on, certain feed additives that worked really well at one given herd may stop working, or the other way around. Feed additives that didn't work so well can suddenly become very effective. So we take a fresh sample every 3 months, and of course, we don't need to sample the entire herd. 15 cows from the herd contain enough material to be a good representative of the herd. So in a very routine process that is done on site, it can even be done by the farmer themselves. They can collect small samples of the rumen fluid from 15 cows, sending it to sequencing, and then based on this data, we can provide the recommendations, and this is being revised every 3 months.
A couple of things. One is, because whilst there's the seaweeds, the 3-NOP that was recently approved by the FDA here in the United States, and a number of other ones that are currently available or close to being available, there's also many other molecules, substances, plants, entities, whatever you want to call them, that people are investigating as a feed additive. And do you need to have real life data of how that additive will affect the microbiome and hence the emissions? Or is there, by understanding the chemistry or the biology of that substance, you can feed that into the model pre actually having to do any real trials?
So because our approach is entirely data-driven, it's basically very easy for us to onboard new additives. And the FDA is now in the process of basically removing obstacles and making the process of approving additional feed additives much easier. So we expect many new feed additives to be introduced to the market. And we have several dozens of farms where we can actually test the effectiveness of these feed additives. And then just by collecting the effectiveness data and matching it with the microbiome samples, then our system is basically trained for these new feed additives. So it's very easy to include new feed additives. And this is, as I said, this is based on the detection of data patterns of significance and predictive capability. In contrast to understanding the mechanisms and so on, that is more a biological approach.
And you mentioned you only needed 15 cows from a herd. Even if that herd is a couple of thousand cows, 15 cows will suffice. Because the reason I'm digging further into this is that there's a number of people, many people I've spoken to who found great variation within the herd. When I spoke to Itzik Mizrahi from Ben Gurion University, he had cows in the same herd on the same diet, and some were producing 100 grams of methane a day, some were producing 500 grams of methane a day. And likewise, there's trials that have showed that animals that have come from different locations have significantly different even emission levels, even though these groups have been on the same pasture for substantial amounts of time. So but you would also expect that if you put an animals in confine or in proximity to the other, that there would be crossover. Of course, the way they eat, the way they move, the way they interact with each other, there would be crossover, and the microbiomes would change because of who the other animals around them are. So I'm just trying to understand that variation that we know exists and the similarities, because they also say that roughly 60% of the microbiome is what they'd call the base microbiome, which is very consistent across all the animals, and then that other 40% is the bit that changes. So I'm just trying to understand why there is some variation, but there's also consistency.
Yeah, so first of all, every cow, it's a being on its own. It has different microbiome and different behavior and so on. However, there is also a very strong herd level common basis. So the microbiome, although it differs between different cows, is pretty homogeneous along the cows in the same herd, or to the very least, much more homogeneous than compared to other herds. And you can actually see the effect in the data that we've collected. We've witnessed, for example, that 3 farms in the same village, comprised of the same kind of cows, buying from the same vendor, of course, experiencing the same weather, reacted completely differently to several feed additives that were introduced. And the reason may be the origin of the cows, as you just said, cows that were born on one certain conditions may evolve different microbiome than other cows, or some pathogens in the herd, or who knows. But the fact of the matter was that their reaction to the feed additives was very different, meaning that although the farms were very close, farmer number 1 would have been paying for this expensive feed additive and didn't get any effect, whereas their neighbor would see a huge surge in the yield. Now, some people are used to think that this is mainly due to the genetics of the cow. However, what we have shown is that actually it is the microbiome which is the most dominant. So, of course, different cows may react differently, and in the same herd of 1,000 cows, you may have 20 that would completely show immunity to some feed additive, when their peers would be very much affected by it. But on average, statistically speaking, the biomarkers on the herd level that we can detect are the most dominant factor with respect to predicting the efficacy of these feed additives. And all the details, of course, appear in the white paper that we released recently for those who are interested in more details.
It's the microbiome is it's such a complex and dynamic thing, and all that goes back to and then how that relates to the actual genetics of the animal. And there's this is somewhere that researchers, as far as I understand, are only really scratching the surface of how much of the microbiome is influenced by the actual genetics of the host animal, and how much is environmental, and how much comes from the early life. Is there anything that you've come across in that space to sort of further understand how that all fits together?
So you are correct that there are many interesting interplays between the microbiome and the genetic of the host and the history and so on. However, as I said, we are not trying to model or explain the biological mechanisms. Instead, because we are taking this purely data-driven approach, we are focused on finding patterns of statistical predictive capability, meaning that we are working very hard to predict what's going to work, what's not going to work, and so on. And the explanation, I agree that it's very interesting, but this is what we leave to our colleagues who are, that this is their field of expertise. We are an AI company. We are a data company. The company is a spin-off of MIT based on, a decade's work in the field of AI and data. However, we are, of course, very much excited to collaborate with researchers in the field of microbiome and genetics that can tell the story, so to speak, explain why the patterns that we detect are actually correlated or even causing the behavior that we observe.
So what's next in terms of this technology? Is it something that someone can currently go and use, or is it still in development? And what's next in terms of having it available so people can use it?
Yeah, so as I said, we spent a couple of years proving that the technology works by collecting this huge amount of data and deploying it in a couple of dozens of farms. We were also part of the Dairy Farmers of America accelerator, working with the DFA's leadership, their nutritionist and veterinarian, in order to validate the concepts underlying our product. And quite recently, we started working commercially, starting in Wisconsin and California, but looking to expand throughout the entire US. So yeah, commercially available, open for business. We started by focusing on dairy, but we've also started proving that it works also on beef. And very soon we will also start serving customers in the beef and cattle segment.
And it's currently just the United States, or is there and what's the thinking on Europe, Australia, South America, Asia,
First,
Africa?
yeah, yeah, so first customer currently are in the US, but we are already speaking with prospective customers in the geographies that you've mentioned. I'll actually be presenting in Australia in a large dairy and beef conference later this year. Also speaking with potential partners in New Zealand and Europe. So yeah, this problem is, as I started, using AI to decipher the information that is encapsulated in the microbiome enables us for the first time to basically take this very traditional work process and make it tailor-made to every farmer and dynamic. So I think that this is something that can really disrupt the business. In terms of numbers, we can triple the efficacy of these feed additives. This is actually the bottom line result. So imagine the billions spent in developing and procuring feed additives can potentially be tripled in their eventual impact. So this is our contribution to this process.
Wonderful. Before we go, is there anything else that you'd like to add that we haven't already discussed?
No, I think that, we pretty much covered most of it. And we welcome any potential partner to contact us and join us in this journey. Basically, the great thing, to my, in my opinion, about what we do is that we bring value to the entire value chain. So consumers get healthier milk. We even show that somatic cell count decrease and cows' well-being actually improves. Farmers make more money, so they make more product at lower costs. And all this magic is coming basically from energy that is being diverted back to the system at the expense of the methane that is not being released to the atmosphere. So, everybody wins, and this is why we find it so exciting.
Wonderful. That win-win is such an important thing to have, because without that, getting everyone on board is much more challenging.
Yeah.
Yaniv, thank you very, very much for joining me today.
Thank you. Thank you for having me.
You've been listening to The Ash Cloud with me, Ash Sweeting, in conversation with Yaniv Altshuler, recorded in California in June 2024.